PCR is a technology that is used to amplify a small amount of DNA into a large sample in a short period of time. There are 3 steps. Step 1, the DNA is denatured by heating up the sample to 95 degrees Celsius. At step 2, DNA primer moves into the 3' end of the DNA when the system has been cooled down to between 55 and 65 degrees Celsius. And finally, step 3, it is heated up again to 72 degrees Celsius, where DNA polymerase from a thermophilic bacteria that lives in geysers moves into the primer on the DNA. This polymerase is called a Taq polymerase and forms a new DNA segment. This process is repeated several times to produce over 1 billion DNA segments after just 30 cycles. Another type of PCR is quantitative PCR, or qPCR. This works in pretty much the same way as regular PCR, however it has a probe at the 5' end of the DNA segment that is added at the same time as the DNA primer. This probe has a fluorescence, and when the Taq polymerase hits the reporter end of the probe, it releases the fluorescence so as to know how much DNA is being made at each cycle.